Hi, everybody. Welcome. Today's lecture, we're going to be talking about skeletal muscle structure and function and kind of its application as it relates to exercise and sport performance. Okay, so to kind of get us started, we want to first introduce some of these anatomical structures. So here we can see a muscle and it's split kind of in a cross section through the muscle belly. And so I want to kind of take a look at some of these anatomical elements. So the first thing I want to address are the connective tissue layers. Okay, so the most outer layer, the epimysium, let me use my little marker here. The epimysium, epi means upon, and the epimysium is going to lie upon or on top of the entire muscle. Okay, so the epimysium is made of a dense connective tissue that's going to eventually form the tendon of the muscle at the end. Okay, the second layer is known as the perimysium. Peri means around, and each perimysium is going to surround each of these muscle bundles, which these little muscle cell bundles are known as fascicles. Okay, so or fasciculi. Okay, so a fascicle is a bundle of muscle cells, which you can kind of see here. And then the innermost layer of these connective tissue linings is known as the endomysium. And an endomysium is the connective tissue that surrounds one singular muscle cell. Okay, so here you can see that connective tissue as it kind of surrounds one single cell, and that is continuous with the cell membrane. Okay, so again, if we go from inside to outside, we have endomysium, we have perimysium, we have epimysium. Okay, and again, all three are made of connective tissue layers that kind of keep things separated and keep things connected. Okay. Additionally, we identified the fascicle or fasciculi as our bundles of muscle cells, which you can kind of see there are several fascicles shown here. A muscle fiber is the same thing as a muscle cell. So again, you can use those words kind of interchangeably. If we say muscle fiber or if we say muscle cell, we're talking about the same thing. And then lastly, myofibril, which I'll talk more about kind of in these upcoming slides, are these individual kind of proteins that are going to kind of interlay with each other in order for us to create muscle contractions. So we'll talk more about that here in these upcoming slides. Okay, additional components to kind of take a look at here. So here you can see a muscle cell again, kind of broken down. You can see these bundles of muscle fibers now in kind of a 3D formation. So again, each of these cylinders would be a fascicle. Okay, and within each of those cylinders, we have many myofibrils or many protein filaments that are going to work together in order for this muscle contraction to occur. Other things to note in this picture as we're kind of adding to the story, we have something called sarcoplasmic reticulum. And sarcoplasmic reticulum is like these little, think of them as like storage bins, and they're going to be storage bins that are full of calcium. And calcium is going to play an important role when we talk about how muscles contract here coming up in just a couple minutes. We also have these T-tubules or transverse tubules. So you can see the term up here. And these T-tubules are kind of these invaginations into the cell membrane. Okay, so they're like a little ditch. Think of it as like a little ditch. So if here's my cell membrane, it's going to be like a little hole that kind of goes across the surface. So again, if this box is one cell, I would have one T-tubule right here in the middle. So it's like a little ditch where I'm going to send an electrical signal. We'll talk about the importance of that kind of coming up here. Let's see, what else do I have? Sarcoplasm. So sarcoplasm is the same thing as like the cytoplasm of the cell. So this is going to be like that gel-like fluid for which we're going to have the ability to have different reactions occurring within. Okay, so again, we've identified sarcoplasm, transverse tubules, sarcoplasmic reticulum, and again, we can see these myofibrils, which are protein filaments, and we'll talk about the different types of myofibrils here coming up. Okay, so again, myofibrils, we said, are kind of these protein filaments or contractile elements within the skeletal muscle. And these are long strands that can be subdivided into smaller units known as sarcomeres. The sarcomere is kind of like your most basic contractile unit within the muscle cell itself. Okay, so we're going to have many sarcomeres within one single muscle cell. So let's have a look-see here. Okay, so here you can see one single muscle cell or one single muscle fiber. And within this muscle fiber, we have many of these protein filaments known as myofibrils. And these myofibrils are going to create singular sarcomeres. So again, if I'm looking at this muscle cell itself, I might have like 100 different sarcomeres kind of broken up here. And again, we'll kind of look at some more pictures as we kind of zoom in. So we're kind of going from the big picture and kind of zooming in to get that microscopic view now. So a little bit more about the sarcomeres and kind of what's up with them. So skeletal muscle are also called striated muscle because they're kind of striped in nature. And the striped appearance that we can see, and I've got an image that looks at a skeletal muscle under the microscope so you can kind of see the relationship. But basically we have these like light and dark kind of stripes. And the gig with this is that basically it has to do with the overlap of these filaments or of these myofibrils within that singular muscle fiber. Okay, so we have thick filaments and we have thin filaments, and the way they overlap will create dark and light striations. Okay, as we mentioned, the sarcomere is the most basic contractile unit of the muscle, and we'll take a look at that here coming up. Okay, so here you can see that striated appearance. So this is like one muscle cell, like this long cylinder chunk. And same thing up here. You can see one single muscle cell in this configuration. Again, striped in nature. The light stripes mean that there's only like one layer of protein filaments, and the darker stripes tell us that there are two layers in that particular area. So again, when we think about the organization of the skeletal muscle, it's very, very nice. It's very well organized. And again, that's going to allow us to produce the greatest amount of force and give us the greatest potential for strength based on this microscopic arrangement. Okay, so now we're zooming in and now we can take a look at what a sarcomere looks like. Okay, so this, from this edge to this edge, is one sarcomere. So I'm going to circle this for you. I'm really getting into the marker today. So this shows you one sarcomere or one contractile unit. And other things to note, as I said, there are two different types of myofilaments or two different types of proteins that we're interested in. These blue proteins here are known as the thin filament or the actin molecules. And this dark red here in the middle is going to be our thick filament or our myosin filament, okay? And the interaction between the two of these is what allows us to create force or allows us to have muscle contraction, okay? So in order for us to produce force, we are going to basically slide those blue pieces towards the middle. So we're going to have them kind of start in this resting position. We're going to push them closer together so that those little blue edges butt up next to each other. And that's the shortening of the muscle, or that's the concentric contraction that muscles can go through. Okay, as I alluded to, the sarcomere contains two different types of proteins. We have thick filaments and thin filaments. Thick filaments are made of this myosin configuration, and these thick filaments have a globular head that's going to protrude out from them and form what's called a cross bridge. Okay, so this thick filament is going to have a lot of these globular heads. Actually, the more globular heads I have, the greater my potential to produce force, because it's through the binding of my globular head with the thin filament that's going to allow me to produce force or allow me to create strength within that muscle contraction. Okay. The thin filaments, also known as the actin molecules, are thinner strands, and we'll take a look at what those kind of look like. But one distinguishing characteristic is they have binding sites for the myosin to kind of grab a hold of them. They also contain additional protein molecules that are troponin and tropomyosin. And we'll talk about what they are going to do here in just a couple minutes. Okay, so a couple things to point out here. Again, here's one example of a sarcomere. So from kind of this bluish gray strand to the other bluish gray strand is one sarcomere or one contractile unit. Now you can see this green strand here is our thin filament, and then in the middle you can see our myosin, our thick filament. Here, these little yellow lines represent the myosin cross bridges, which we'll talk about in more detail. But notice with this thin filament, it's kind of like a twisted double strand of proteins. So again, it's the interaction of these two molecules that's going to allow us to produce force within muscle contraction. Okay, so here in this slide, you can see as we kind of zoom in, here in the green is that thick filament with a myosin head, and here in the purple, you can see that double strand of that thin filament or that actin molecule. Other things to note when we look at this actin molecule is this troponin and tropomyosin. Now, troponin's job is to serve as a binding site for calcium. So when calcium comes and binds with troponin, that causes tropomyosin, which is like this pink strand, to move out of the way. And when tropomyosin moves out of the way, it uncovers the binding sites, allowing this myosin head to connect to the actin molecule. And again, when those two things connect, that's what allows us to generate muscle force. Also notice that there's ATP or ADP kind of involved here. And essentially what we want to show with this is that each time the actin and myosin interact, we are going to expend energy, which is why muscle contraction costs us calories, right? So again, the harder the exercise or the more work I'm doing, the more muscle contraction I'm doing, the more energy I need, or the more calories I burn, or the more ATP I'm kind of utilizing. So other kind of key terms to note when we talk about muscles and kind of their interactions and kind of how this all fits together, we have an alpha motor neuron, we have a motor unit, and we have a neuromuscular junction. Okay, so the alpha motor neuron is like the neuron that's in charge of causing muscle contraction for each individual muscle. Okay, so one alpha motor neuron is in charge of many individual muscle cells. And when that alpha motor neuron sends an excitatory signal, it causes the contraction of each of those muscle cells that it's in charge of. Okay, so think of the alpha motor neuron as kind of like the boss, and each of those muscle cells is like one of his employees. Okay, now one other thing that's important to note here when we think about these relationships is that each muscle cell can only have one boss. So each muscle cell is only going to be controlled by one alpha motor neuron. However, one alpha motor neuron can have multiple muscle cells that he's in charge of. Okay, so the motor unit is one alpha motor neuron and all the muscle cells that he innervates. Okay, so motor units can vary in size, and we'll talk about some of those principles here coming up, but they can vary in size kind of based on the goals of that particular motor unit. Okay, the neuromuscular junction represents the interaction between the alpha motor neuron and the muscle cell that he innervates. So let's kind of set the stage in terms of understanding how we can have a muscle cell stimulated, or how can we have a muscle cell become activated. Okay, so the first thing that we need to do is we're going to have an alpha motor neuron is going to send an action potential or send an excitatory signal. Okay, as that action potential travels down the neuron's axon, it's going to reach the axon terminals. And at the axon terminals, that neuron is going to release a neurotransmitter known as acetylcholine into the neuromuscular junction. And again, that neuromuscular junction is just the interaction between neuron and muscle. And also important to note that it's just like there's like a little gap there. There's like a little space in between the neuron and the muscle cell. Okay, so acetylcholine is going to bind to receptors on the muscle cell. The muscle cell opens gates to allow sodium to come into the muscle cell. As the sodium enters the muscle cell, this becomes depolarized or excited, and that's going to help us to send that signal from neuron to muscle cell. Okay, so again, now that the signal is sent from the neuron to the muscle cell, an action potential will be generated and travel down the T tubules or the transverse tubules of the muscle. This signals the release of calcium from the muscle's sarcoplasmic reticulum and dumps it into the sarcoplasm. Calcium binds to troponin. Troponin moves out, causes tropomyosin to move out of the way, uncovering the actin binding sites. When those binding sites are uncovered, actin and myosin are going to interact, and that is muscle contraction. Okay, so I've got a couple images to kind of show you this in more detail, but now you have kind of the word version of it, and now I want to take you through these images. Okay, so let's start over here. Here up at the top, you can see the alpha motor neuron, and this section down here is the muscle cell. Okay, so the alpha motor neuron has a depolarization, and as that depolarization or exciting signal reaches the axon terminal or the edge of the muscle, or the edge of the neuron, it causes the release of acetylcholine. Okay, acetylcholine is a neurotransmitter that's going to head into this junction, this neuromuscular junction. Now notice that the neuron and the muscle cell aren't actually touching each other. Okay, so acetylcholine is going to bind to the edge of the muscle cell, causing the muscle cell to be excited. Okay, sodium is coming in, causing the muscle cell to become depolarized. That depolarized signal or that action potential is going to travel down these transverse tubules, okay, across the membrane. And as the signal travels down this T tubule, it causes the calcium, which is stored in the sarcoplasmic reticulum, to be released into the sarcoplasm. Okay. Once calcium is released into the sarcoplasm, it's going to come on over to the actin thin molecule, bind to troponin, cause tropomyosin to move out of the way, and actin and myosin will then interact. Okay. This on this side shows us what happens when the muscle contraction is done. Okay, so acetylcholine is going to be removed from the neuromuscular junction through an enzyme known as acetylcholine esterase, which basically think of it as kind of like a vacuum cleaner, right? It's going to kind of clean out that area and get rid of that acetylcholine so that the excitation process stops. Okay. Next, the calcium will be shuttled back into the sarcoplasmic reticulum where it'll be stored, and then the actin sites are going to be covered back up so that the myosin and actin can no longer have an interaction, and the muscle itself, those muscle fibers will kind of spread back out, causing the muscle fibers to reach their relaxation state or position. So again, this just tells you in words what's happening when the muscle contraction needs to stop. So it will continue as long as there's calcium available. And as soon as that signal stops, we want to pump that calcium back into the sarcoplasmic reticulum where it's going to be stored. And therefore, troponin and tropomyosin will return to the resting configuration so that the actin and myosin can no longer have an interaction. Simultaneously, we are getting that acetylcholine out of the neuromuscular junction with that enzyme acetylcholine esterase in order for us to stop the excitation signal that's happening at the level of that muscle cell. Okay, again, this is the exact same process, just a different image. And again, I like this image because it's kind of got those different steps labeled. I also included a YouTube video here if you want to watch an animation of how this all happens in terms of the different steps and the sequences of events that need to occur in order for muscle contraction to occur. Okay, so now we're kind of zooming in. The next step that we want to kind of take a look at is what's happening within that muscle contraction itself. Okay, so again, now we're kind of zooming in and taking a look at these filaments, okay, or these protein molecules within the muscle cell. So here you can see the thick filament. Again, myosin is made up of two strands of proteins that are kind of twisted together, and they have globular heads. And these globular heads are going to project out from that thick filament and have an interaction with that actin thin molecule when those binding sites are uncovered. Okay, so the myosin heads will only interact with the actin molecules when those binding sites have been uncovered. Okay, so again, as we mentioned, the thin filaments or the actin molecules will have really three major components. So actin is kind of the backbone of the filament. Okay, so again, imagine that these are strings of beads and it's like two strings of beads kind of twisted together. Okay, so we'll have individual globular actin molecules that are joined to form a strand. We have two strands, we're going to twist those strands together. Okay, tropomyosin is a tube-shaped protein that twists around that actin molecule. And again, its job is to cover the binding sites when it's in its resting configuration. And then troponin is a smaller protein that serves as a binding site for calcium. So again, how does this all interact? So once calcium is released, it comes and binds to troponin. Troponin's job is to tell tropomyosin to move out of the way. Tropomyosin moves, uncovers the actin binding sites, and that allows the interaction of actin and myosin. Okay, so here you can see kind of what this looks like. So here you can see these like white beads. These big white beads are the actin. And again, think two strands of beads that are kind of twisted together, and that's what the actin molecules look like. Then you can see the tropomyosin is like this thin strand-like protein that's going to cover those binding sites. And then you have troponin, which is a smaller molecule that will serve as kind of a docking point for the calcium to bind onto. And the binding of that the calcium to the troponin causes the tropomyosin molecules to move out of the way. Again, a little bit of repetition here, but looking at the role of calcium as it relates to the muscle fiber actions. So once calcium ions are released from the sarcoplasmic reticulum, they bind to that troponin, that little molecule. The binding of calcium to troponin initiates the contraction process by moving tropomyosin out of the way. So we know that without calcium, muscle contraction cannot occur because we need that calcium in order to open up those sites so that actin and myosin can interact. And the interaction of actin and myosin is what allows us to produce that muscle force. Okay, so then what happens, right? So again, we've talked about actin and myosin need to interact. But in order for us to cause the shortening of the muscle or cause that sarcomere to get a little bit smaller, and again, I would reiterate that the reason that the muscle shortens isn't that any of these molecules individually are getting shorter, it's the overlap of these molecules, right? So again, these protein strands are going to stay the same length. However, we're just going to kind of smash them together so that they're more overlapping when that muscle shortens, okay? So how does this work? We've got four processes here. So we're going to have cross bridge formation. This is when the actin binding sites are uncovered and the myosin head is going to bind on to the actin molecule. Okay, the next thing that's going to happen is called a power stroke. And so that myosin molecule is going to pull that actin forward, causing those thin filaments to slide together. Okay. Then once we've slid them together, the cross bridge will detach and reactivate and get ready for a new cycle. Okay, so again, I'm going to back up so you can see this a little bit easier. So again, myosin binds to the actin, it pulls the actin forward, it detaches, it gets ready to bind again, it pulls forward, it detaches, it gets ready to bind again, it pulls forward. Okay, so again, we're just sliding those thin filaments in as that actin and as the myosin grabs a hold and pulls that actin forward. Okay, this process will continue as long as calcium is present or until those thin filaments butt into each other. So once they run into each other, we've reached kind of a full contraction. That's it. Then they're going to kind of stay in that position and create force in that position, but they're not able to cause movement from that position because they're already like running into each other. Okay, in order for this to happen, we have to have energy. Okay, so we're going to use ATP in order for this whole cross bridge formation to occur. And interestingly, there are different enzymes or different ATPase that's going to determine how fast this can occur. Okay, so again, if I had a fast ATPase, that would allow me to quickly have that muscle contraction occurring. Okay, so somebody that has a fast ATPase is going to be quicker and more powerful than somebody like me who has a slow ATPase, and they're not going to be able to generate that force as quickly. So again, let's take a look at that as it relates to the speed of ATPase. That will determine in part our muscle fiber type. Okay. More to come about muscle fiber type. But here we can kind of take a look at what's going on. So here you can see this cross bridge cycling. Okay. So again, this is going to continue on in a circle. Okay. So we've got the myosin head. It's going to have cross bridge formation. So it connects to that actin. We've got the power stroke where that myosin head is pulling that actin forward. We've got detachment, and the detachment is going to cause ATP to be spent and reactivation of the myosin head so that you're ready to do it again. Okay, so again, once we get to this reactivation phase, we're going to grab another ATP so that we're ready to do this process again. Okay, so what does this look like? So again, here you can see the myosin, here you can see the thin filaments, the actin. When they're in a relaxed position, the actin molecules are not touching each other. Okay, so they're far apart. And essentially what's happening when we contract is we're pulling those actin ends into each other. Okay, so here you can see in a relaxed position, the actin molecules are butting up next to each other as that muscle shortens. Okay. Keep in mind, again, the length of those fibers don't actually change, but the overlap is what allows that muscle to shorten. Okay. Again, I provided you with another little video here that I thought was a good illustration of this cross bridge cycling and kind of how that works and how that kind of all fits together. So feel free to kind of check out that video to get maybe a better visual of all these moving parts together. Okay, so again, when muscle contraction is done, we're going to pump calcium back into the sarcoplasmic reticulum. Troponin, tropomyosin return to their resting configurations, and this will prevent that cross bridge cycling or the binding of the myosin and actin with each other. Okay, so far we've talked about something called shortening, but in reality, there are really three different types of things that can occur from a muscular contraction perspective. Okay, so we have concentric contractions, isometric contractions, and eccentric contractions. And in reality, when we do coordinated movements, we have to combine and mix and match all three of these types of muscle contractions in a coordinated way in order for us to be able to execute these complicated or complex movements. Okay, so concentric contraction is the shortening process, right? So that's the process that we've just talked about, where we're going to have those actin molecules that start far apart. We're going to slide them together so that that muscle is shortening. Isometric contraction, iso means the same, metric means length. Okay, so isometric contraction is when we're generating force, but the muscle length isn't changing. Okay, so think like stability types of things. Okay, so if I were to do a pull-up, for example, I'm going to have an isometric contraction of my abdominal muscles. And again, the reason is that's going to create a rigid core for me so that I can do that motion without having injury, without falling, keeping my body kind of under control. Okay, another example of an isometric contraction would be when you do a wall sit. Okay, so when you do a wall sit, we're generating a lot of force. Like you can do this and try it out and know that it's challenging to do, right? However, there's no change in the muscle length. Okay, so again, isometric, we're looking at that muscle length will stay the same. However, I am generating force. And then lastly, when we think about eccentric contractions, this is the lengthening of a muscle under tension. Okay, so again, the key with eccentric contractions is that the muscle has to be under tension in order for us to gain anything out of this eccentric movement. Okay, so with eccentric contraction, again, imagine that that muscle is already fully contracted. So let's think about like, how do I show you this? Bicep, for example. Okay, so when I get to full contraction, full shortening, I have to also lengthen that muscle out. Okay, and that lengthening of that muscle out, as long as I'm under tension when I do that, that's going to allow the actin molecules to slide apart kind of grudgingly, under force and under control, and that's what an eccentric contraction is. Okay? So how do we decide how much force we are generating? Okay, so how do we know how much force we need? Okay, again, if I wanted to pick up my coffee mug, I don't need to activate every single muscle fiber that I have, right? However, each muscle fiber will either act on a all or none principle. Okay, so that means that that muscle cell is either going to contract or it's not. There's no like large or small contractions within an individual muscle cell. So a muscle cell is either in or out. You're either going to work or you're not. There's no in between. There's no like half days or anything else like that. It's like you're doing it or you're not doing it. Okay, so in order for us to determine how much force or modify how much force we are producing, we can modify how many muscle cells are involved, right? So again, if each worker is either working or not, I can decide how many workers go to work, right? So I can either have 30 or I can have 100 workers, okay? So the number and type of motor units stimulated will be in part determining the generation of force, how much force I'm producing. The frequency of stimulation, so how many signals am I sending to that muscle cell? Okay. The size of the muscle in general, right? The larger the muscle, the greater the force potential. However, you know that we modify that based on how much we need, right? So again, I know from memory of picking up this coffee mug before and picking up things like that, then I need to activate, let's say 20 muscle cells or something along those lines, right? So I know from my memory about how much work I need to lift up this coffee mug. Okay. Muscle fiber and sarcomere length also makes a difference. So again, thinking about like if the muscle is already fully contracted, I can't generate any more force because it's already there. Okay. We'll talk about optimal positioning for the muscle as well coming up. And then speed of contraction, right? Are we trying to move this fast or are we trying to move this slow? And how does that affect how much force we can generate as it relates to those actin myosin molecules connecting to each other? So this is kind of cool. So this shows us the relationship between speed or velocity and our force potential, okay? So I'm going to give you a couple different examples here to kind of explain this force velocity curve, right? I'm going to start with continuing my conversation about biceps, okay? So if I go into the weight room, okay, and I'm going to do bicep curls and I grab 5-pound weights, okay, can I stand up to do this? Like you be able to see me? If I have 5 pounds, I can easily do this really fast. Okay, this is no problem. I can produce enough force in order to do my bicep curls really fast. Okay, if you hand me 15 pounds, that's going to be pretty tough for me. I can do it, but I'm going to have to go slow. Okay, so in this example, we're moving from a low amount of weight where I can move really fast to a higher amount of weight where I have to move a little bit slower. Okay, so lightweight can move a lot faster in comparison to heavy weight when we're on this concentric side. Okay, so with 15 pounds, I could do it, but it was hard. I had to go slow in order to truly get that weight up. Okay, you hand me 20 pounds and I can't get it all the way up, but I can get it here and I can hold it. Okay, so with 20 pounds, I can hold the weight here, but I'm not strong enough to get it all the way up. Okay, so I can produce an isometric contraction, but I'm not strong enough to move that 20 pounds in a concentric way. Okay, so now you hand me 25 pounds, for example. Okay, I'm not strong enough to hold it here, but if you handed it to me up at the top, I can lower it down under control. Okay, so the greatest amount of force that I can produce from an eccentric perspective is kind of up here. Okay, so again, when we think about how this applies to weight, right? So we said 5 pounds, I can move really fast. 15 pounds, I can move really slow because it's harder. 20 pounds, I can hold, but I'm not strong enough to move it. 25 pounds, I can lower down, but I'm not strong enough to even hold it or do concentric contractions with that. Okay, so in this slide, we're taking a look at speed of contraction as it relates to resistance. So essentially, we're looking at this force velocity curve, but only considering what's happening on that concentric side of things. Okay, so here you can see resistance and speed are inversely related. Okay, so the more weight I'm trying to move, the slower I have to move it. The lighter the object is, the greater the force in terms of velocity. Okay, so again, these are inversely related, which makes sense. If I try to move a light weight, I can move it quickly. If I try to move a heavier weight, I have to move it more slowly. Okay. Another factor to kind of take a look at here when we think about muscle contraction is something called the length tension relationship. And essentially what we're talking about here is the interaction or the overlap of actin and myosin and how much movement we have to have in order to get that actin molecules kind of butted up next to each other in a full contraction. Okay, so the length tension relationship tells us that there's an optimal length of each muscle fiber relative to its ability to generate force. So when we think about how this kind of applies, I'm going to show you on the next slide and kind of explain what I mean by this. So here you can see the length tension relationship. And essentially what we're saying is there's an optimal level of overlap where the actin, the myosin heads can grab onto the actin. The greatest number of myosin heads can grab onto the actin in this position, okay? We can also have too much overlap. So again, if we think about like the bicep, which I kind of talked about earlier, when you think about the relationship here, when I'm in full contraction, which is shown here, there's too much overlap. I can't create any more force or because there's too much overlap. Okay, I can also have this position where no cross bridges can attach because the muscle is too stretched out, right? However, kind of that optimal length is going to be kind of in the middle of that range of motion, which is going to be here, which is going to allow us to produce the greatest amount of force. So kind of moving forward and talking about, we talked about the alpha motor units, and the motor units had to do with one single alpha motor neuron and all the muscle cells that innervated. We also talked about ATPase and how ATPase can have varying speeds. And essentially we can classify these different muscle cells based on the ATPase speed. Okay, so muscle fiber types, we have 3 different muscle fiber types. Type 1, type 2A, and type 2X. And how that relates to the motor unit is that every muscle cell within a motor unit is going to be made up of the same type of muscle fibers. Okay, so again, if I am the alpha motor neuron, I might be in charge of 12 muscle fibers or muscle cells, but they all have to be the same type. So they're all type 1, or they're all type 2A, or they're all type 2X. Okay, so again, within one single motor unit, I have all of the same types of muscle cells. Okay, now also appreciate that these fibers are going to be kind of like intermixed, right? So they're going to be kind of mixed together. However, who's in charge of them is going to match up based on the type, right? So again, an alpha motor neuron can be in charge of all these type 1 cells. A different one can be in charge of these type 2As, and they're going to kind of be mixed together for us. Okay. So what about this information? So again, type 2 fibers in general are going to be your fast twitch fibers. So again, we have 2A and 2X. So type 2A are going to produce more force and they're going to be faster. So they're going to be like good for short, high intensity, like endurance events. Versus type 2X, these are our true anaerobic, true fast twitch fibers. These are going to be important for short, explosive types of exercise. Okay, so again, these are our greatest force producers. Type 2As are going to be kind of like an intermediate fiber, and type 1 fibers are going to be those slow twitch fibers or endurance based. Okay, so here you can see a little bit about each of these fiber types. Okay, so again, type 1 is going to be slow twitch. Type 2A is going to be kind of like an intermediate fiber, and type 2X is going to be fast twitch. Okay, so again, looking at some of these different characteristics. So fibers per motor neuron. So again, type 2A is going to be the smallest, type 2X is going to be the largest, and again, type 2, I'm sorry, did I say that right? Type 2A is going to be intermediate in nature. Okay, motor neuron size, small for type 1 and large for type 2X, et cetera, et cetera. So again, thinking type 1 is more slow and type 2X is going to be our fastest. Okay, so when we think about fiber type and athletic success, unfortunately, we can't determine which type of muscle fibers that we have. We can only make the most of what we have, right? So again, this would help us to understand that different athletes are going to have different potentials and different strengths and weaknesses as it relates to different types of sport, right? So some athletes are going to have a lot of endurance and be good at kind of that longer, slow type of exercise, and those athletes are going to have a lot of type 1 fibers. Okay, they're going to have a lot of that slow twitch fibers. Athletes that are powerful and quick and can generate a lot of force, generally they can gain muscle mass a little bit easier. They're going to have more of those type 2 fibers. Okay, so again, you know, when we think about this as it relates to athletic success, just consider this as one component of our potential, right? Certainly, this isn't the sole predictor of success in sport. You know, cardiovascular function, motivation, training habits, all of these factors are going to also play an important role. And again, I think our jobs as coaches isn't necessarily to be focused on creating, you know, all Americans out of all our kids or state champions out of all of our kids. However, our job is to help them reach whatever their potential is, right? And again, that can vary from kid to kid in terms of, you know, where is your ceiling? So I think our job is to get the most out of them, regardless of, you know, kind of what their genetic background looks like in terms of setting them up for success. Okay, so again, we said that with these motor units, right, it's an alpha motor neuron and all the muscle cells it innervates. And as we mentioned, each motor unit will either be fast, intermediate, or slow in nature. Okay, so our large motor units are going to have greater than 300 muscle cells, and they're usually those fast oxidative, so like a type 2A or a type 2X fiber. Okay, so again, those fast twitch fibers. And then our small motor units are going to be less than 300 muscle cells, and they're usually going to be slow oxidative. So those type 1 fibers that are going to be more kind of endurance based. So when we think about recruitment, okay, we're talking about the order in which these muscle cells will be activated. Okay, so when an alpha motor unit is activated, all the fibers in that unit are going to contract. Okay, so again, everyone in that unit will go to work and do their full job, right? So the way we monitor or manipulate force production has to do with how many alpha motor units are activated. Okay, so activating more motor units will produce more force. And initially, we need to activate type 1, then type 2A, and then lastly type 2X. Okay, so we're going to activate these from slow to intermediate to fast. Okay, so our slow twitch fibers are going to be used most frequently. And again, we think about like activities of daily living and that sort of thing, like these type 1 fibers are going to be used all the time. It would be rare for us to use these type 2X or these fast twitch fibers unless we intentionally try to, right? So even when, you know, your average exerciser is doing exercise, right? If you're going for a walk, a bike ride, whatever, we're really still only activating those type 1 fibers, right? So again, we're only going to activate these type 2X fibers if we're really specific and we're really purposeful about what we're doing. And in order to activate these, we've got to get going with high intensity, high power, high velocity type of work in order to get your most bang for your buck out of these. This is cool. This just shows you kind of the relationship between all three. So here you can see motor unit 1 is in green, motor unit 2 is in yellow, and motor unit 3 is in purple. Sorry, this is a little bit hard to read here. But notice that those motor unit 1, those slow oxidative or those type 1 fibers are small. So here you're kind of looking at a cross section of the muscle, and notice that they're very small in nature. So we will activate those first. Then second, we might activate those intermediate fibers. Okay, so those type 2 intermediate fibers will be activated second, and that's going to be here in yellow. Again, they're kind of intermediate in size. And then lastly, to get to those type 2X fibers, or those fast twitch fibers, those are going to be the largest muscle cells that you have, and we're going to activate them last. Okay? So when we think about, generating muscle force, right? So a couple different terms to kind of keep in mind. We think about muscular strength. We're talking about what's the maximum amount of force that I can produce in a short window, right? So it might be a 1 RM. How much force can I produce in 1 rep, right? Or how far can I throw the shot put 1 time, right? So those are going to be acts of muscular strength, right? Muscular endurance, however, we're looking at multiple repetitions at a submaximal intensity. Okay, so again, maybe with this, we want to do like number of push-ups completed. Okay, so again, assuming that the athletes that you're working with can do more than just a couple, we're looking at this as a measure of muscular endurance. How many reps can they complete at the submaximal intensity? Okay, and then when we think about muscular power, we're also considering what is the time that it takes to do this thing. So it's not enough to just do it with force. We want to also do it with speed, right? So thinking about activities like snatch or power clean, things like that, where I want to move a large amount of weight, but I want to do it very quickly as well. Okay, so that's an example of muscle power. Again, these are different types of contractions or different types of force potential, you could say, within the muscular system. And again, when we think about who's going to contribute to these from a muscle fiber type perspective, certainly these muscular strength requirements will come from the type 2X fibers. Muscular endurance are going to be more of the slow twitch type 1 and type 2A. And then muscular power is going to be more type 2X as well. When we think about muscular fatigue and muscle force declines, right? So what is fatigue? We're looking at our decline in our ability to generate force. Okay, so again, when we think about this and how this might apply to these different scenarios, it's important that we think about the energy systems, honestly. So again, with muscular strength, what is our reason for fatigue, or our potential for fatigue? Like what would set one athlete up for success from a muscular strength perspective and not the other? We could consider things like muscle fiber type or muscle fiber size as well. Those are going to be huge in terms of determining how much force I can produce in one given repetition. Okay, so again, for muscular strength and muscular power, I would say our potential for fatigue has to do with the size and the genetic makeup of that muscle fiber. When we think about muscular endurance and fatigue, again, now we're kind of taking a look at, are they building up lactate? Are they building up hydrogen molecules? Are they depleting themselves of that ATP phosphocreatine system? So again, I think all of those factors are important for us to understand when we think about fatigue and how it affects this particular system as it relates to sport performance. All right, so I think we'll stop there today. Please reach out if you have any questions. Hope you have a great day.